right, well, welcome back. I am reporting to you live and in stereo from my kitchen. We're going to be returning to our regularly scheduled IB Bio program. To do that, we're going to start with topic 1.5, which is the origin of life. But before we get to that, just a couple of announcements. Please do update that beetroot data. I think we only have data from group 5, so we need that from the other groups in order to run our statistical analysis. So dig deep in those backpacks, find those scraps of paper where you guys wrote that down, and head over to that shared Google file, that sheet that I asked you to upload it on. Also, check the choice board that's on Google Classroom for more activities related to this content and some helpful videos to help you along the way. All right, so onward. So think back to a couple weeks ago when we were talking in the introduction of cells, and what is cell theory? And I know I've asked you guys some of that so many times, you're probably going to hear that question in your nightmare, but go ahead and let me know what you think. What parts of that do you remember? All right, so cell theory, just to recap, is that all organisms are composed of one or more cells, that the cell is the smallest unit of life, and that all cells come from pre-existing cells. And so if you were, keen on that, you might have been like, okay, well, if all cells come from pre-existing cells, where did that first cell come from? And what we know is that the oldest cells are estimated to be around 3.8 billion years old. So that's your great whatever grandfather. But where did that first one originally come from? So people have been asking this question for a really long time, and up until a couple hundred years ago, they were like, okay, we know exactly how this goes. See, life just spontaneously arises. And so this was the idea that life can come from non-living matter. So they used to think that maggots would just magically appear out of nowhere on meat, or that mice would just randomly show up. So this is what people used to believe up until, again, a couple hundred years ago, that life would appear just randomly. So it would happen all the time. So for example, clams would arise from sand spontaneously. You can go out into a beach and see this phenomena for yourself. And they're like, clearly this is how the world works. And there used to even be recipes for different kinds of organisms. So for example, if I wanted to make a mouse, I would just put one piece of soiled cloth, add some wheat, and wait 21 days, and voila, I have some mousies. Or if I wanted to make a scorpion, I would just take a piece of basil, stick it between two bricks, and put that in the sunlight, and then blammo, I have a scorpion that would just spontaneously generate. But being the good scientists that we are, can you think of some flaws in this experiment using what you already know? So what you probably already guessed is that this is an open system, and those scorpions and the mousies, they didn't just spontaneously generate. This was an open system. They never closed it off, so those creatures just walked in from somewhere else. And to fix that, what we have to do is to seal the system and run the experiment again and see if we can get organisms to spontaneously generate. And one man did this, a French scientist by the name of Louis Pasteur in 1859, for whom the Pasteur Institute in Paris is still named. And so he does this experiment where he has one system that is open and one that is closed, and only the open system grew bacteria that was, presumably floating in the air, so some of it gets in. And so this demonstrates that cells only come from pre-existing cells. So now that I told you that, let's take a look more closely at how this experiment was actually run. So he uses these swan neck flasks. And in these flasks, he has this nutrients broth. So all it is like pizza and McDonald's for bacteria. They love it. So he heats both of these flasks up, which kills bacteria. So he's now sterilized each of these flasks. But to one, he just leaves it alone because bacteria is not going to crawl up this neck here, so it really can't get in if it's floating around in the air. But to the other, he removes the neck, exposing it to the open air. And sure enough, the one that stayed sealed, no bacteria was present, and the one that was open, it started to grow. So what this suggests is that bacteria in the air colonized the broth, and he's thus disproving spontaneous generation as a means of generating organisms. So just to review, spontaneous generation is the old idea that life generated from non-living material, and that this was disproven by Louis Pasteur in 1859, where he sterilizes two flasks with nutrient broth, he exposes one to air while the other one stays sealed, and only the one that was exposed to the air, which has bacteria in it, grew that bacteria in that broth. Okay, so if cells, that brings us back to our question, if cells come from other cells, where'd that first one come from if things don't spontaneously generate? And here's some uncomfortable truth. We really don't know, at least not for sure, but we do have some theories. And so that's where the abiotic origin of life theory comes in. And so I know I just got done telling you that organisms don't spontaneously arise, but they're saying that this happened at least once. And so before I tell you that story, we have to talk about what we do know. So they're saying that cells may have come from abiotic materials that were on early Earth. And the Earth 4 billion years ago was very different from the Earth that we live on today. So for one thing, there is no oxygen in our atmosphere. It was a completely anaerobic environment. And secondly, there was really no atmosphere to speak of. So meteorites and comets, they could come in kind of unscathed a lot of the time. And it was a pretty hostile environment. So it's kind of like a fiery hellscape here. A lot of volcanoes, a lot of comets hitting the planet all the time. Now, can you think of any organism that really loves to thrive under anaerobic or hostile conditions? So one of the 3 domains that we have talked about, which one of those loves to thrive under anaerobic hostile conditions? Okay, so if you said archaea, you are completely correct. I should also mention that even though there wasn't a lot of oxygen, there was a lot of methane. And so if you remember those methanogens, those were archaea. And again, those are the ancient forms of life. They were the first ones that they think that formed. So even an archaea, which is a prokaryote and it's pretty simple, has a lot of parts to it. You guys know this because you've drawn them a million times. So what are the steps to making an archaea? So what they think is that there were 4 things that needed to happen in order for an organism to form originally. So this would be a sequence of events that potentially, we don't know, potentially occurred to go from those abiotic chemicals, stuff that you can hold in a bottle, to form something that is biotic and living, something that has those functions of life, those 7 functions, those Mrs. Hangs, you remember? So how do we go from chemical soup to living organisms? So the first thing that has to happen is that you need to have some production of simple carbon compounds. And so what we know, again, about that early Earth is that there were chemicals present in abundance on this planet. So one of them is methane. Again, those methanogens love to eat this stuff. There's water on our planet. And again, when they're looking for life, they're always looking for water. This is why we also had a lot of hydrogen gas and ammonia present on the ground in our atmosphere on early Earth. And if you start looking at these, the carbons, the oxygens, and the ammonias, you'll notice that we have these atoms quite frequently, a lot of biomolecules, and so we need these guys to start forming simple molecules. We already have the elements here. So then the question becomes, can organic molecules form spontaneously from these starting ingredients? And yes, they absolutely can. And so it turns out that there were these couple of mad scientists, Miller and Urey, who they go out and they get water and ammonia and methane and hydrogen gas, and they stick it in a flask. And in very mad scientist fashion, they apply an electrical charge, and what was produced was kind of amazing. So in that mix, when they did that, out came more than 20 amino acids, including the amino acids that are the building blocks of all of our proteins. In addition, they found ribose sugar, which you may remember from such lectures as nucleic acids, right, in 2.6. So that's the sugar that's in RNA. They also had bases like adenine, which you might remember from submolecules as ATP. That's the A in ATP, which we use for energy. It's also the A in the A's, G's, T's, and C's from DNA, and the same A that you find in RNA from the A's, G's, and U's, and C's. So these bases just showed up when they took those ingredients that they know are on Earth and shocked them. It just created it. So other fun fact for you is that these organic compounds are found in meteorites. So this is one of the more famous ones, the Murchison. So this is a class of meteorite, this carbonaceous chondrite. And so this rock is dated to be 7 billion years old, and it was found in Australia, and it contained multiple amino acids like glycine, alanine, and glutamic acid. It too had some of those ribose molecules, and multiple meteorites show these. And so these are space rocks that are billions of years old. I assure you, nobody out there was like sticking stuff in flasks and shocking them. So these organic molecules form on their own, and it's not so infrequent that we can find them on rocks that fall into our atmosphere. All right. And so just a reminder that meteorites hit planets all the time. So I know this is a picture of the moon, and I know that the moon is not a planet, so don't send me hate mail about that. I'm only showing you this to show you that this moon is being hit quite frequently by meteorites. So all of these large circles here are from meteorite impacts, this crater surface. So it happened all the time to the moon. It happens all the time to Earth. These meteorites are out there carrying these organic chemicals, and they're flying into planets all the time. So those organic molecules, those simple ones, are kind of forming out there by themselves all the time. So the second thing, even when we're starting with those simple building blocks, is the formation of carbon polymers. So they think that maybe a site for these formations of the carbon polymers was at the deep sea vents. So this is like deep at the bottom of the ocean where the magma is coming out of the Earth's crust. It's really hot, which is giving a lot of energy, and you have reduced inorganic chemicals like iron sulfide that are going to really encourage any molecules that are around to start forming chemical reactions with each other. And so it could provide energy to form things like fatty acid chains or your nucleic acid polymers like we have here. So again, these are just bases, and this was the ribose. So this is going to show up just randomly by itself through chemical reactions on Earth, and so it just needs a little push to start forming these polymers. So this would be like a nucleotide polymer, or this would be your fatty acid polymer. So we have methane just sitting around by itself, and it would just need to be in this like heated environment to want to start forming links to other molecules. And so same with the amino acids. So these individual blocks are sitting around, and if we just encourage these guys at the end to like lose some electrons, they can grab onto their neighbor and start forming chains. So this is just sort of what these deep sea vents look like. So again, these are really hot waters, and again, archaea love places like this. So that Taq polymerase that we were using in PCR, the bacteria that makes it is an archaea, and it comes from an environment just like this. All right. So once we form polymers, the thing that we need to form now are membranes. So remember that amphipathic molecules, of which phospholipids are, so this is your phospholipid molecule. And remember that amphipathic means it has both hydrophilic and hydrophobic properties. These guys are going to want to race to form a lipid bilayer all by themselves, right? Because this hydrophilic head, remember that phosphate has negative charges, and it's going to want to race towards water. It's water loving, it's hydrophilic. And whereas these tails, because they don't have any charge, are going to get pushed around by the molecules, so they're going to automatically form these little vesicles by themselves without any other energy coming in. You guys saw that for yourselves in the detergent milk dye experiment that we did in class. All right, so the last thing is that we need to have some sort of development of an inheritance mechanism. So currently, all living things, we have genes encoded by DNA, which are then transcribed by RNA, which is then translated into protein. But just a reminder, remember RNA has its own set of 4 nucleotides too, the A's, G's, U's, and C's. And RNA can catalyze reactions just like proteins can. So yeah, we have our DNA molecule, but this is what RNA actually looks like. It will form its own base pairs, and again, it has its own nucleotide sequence. And then this suggests that RNA was the early molecule of inheritance. There is a link in the choice board going into more detail about the RNA world, and it's really cool. You should check it out. So, but let's take a look, for example, at the ribosome. So we've been drawing ribosomes as little dots, but they actually come in 2 halves here, so a large and a smaller subunit. And remind me, what do ribosomes do? Okay, so ribosomes synthesize protein, right? And so in this picture, we have a large and a small subunit of your ribosome. And the green part is the part of the ribosome that is protein. But that blue and gray stuff here is RNA. So the majority of your ribosome that's making protein is actually made of RNA, on top of which the RNA is actually doing the reactions. It's actually catalyzing the reaction itself. It's not protein. And what's more, the stuff that's moving the amino acids are another kind of RNA. These are tRNAs, transfer RNAs, that are carrying around amino acids on them. So that just gives a lot of evidence or suggestion that maybe the RNA world theory is true, but that's still up in the air. All right, so evidence for the theory that life formed from abiotic forms is that all organisms use the 4 bases in DNA. We all have the A's, G's, T's, and C's. We all use the same 20 amino acids, and we all get those amino acids from a 64 codon sequence. So this is the same in every single organism. So this would be the code that you get from RNA. These are your A's, G's, U's, and C's, and these are the 20 amino acids. So because we all use the same system, it suggests that we have a single ancestor from a really long time ago, about 3.8 billion years. All right, so just for review, right, there are 4 steps in what they think happened that would be required for life to arise from abiotic chemicals. And so the first part is production of simple carbon compounds like sugars and amino acids, and this was demonstrated experimentally by Miller and Urey that this can occur with just some starting ingredients like what you had at the beginning of Earth's history. After that, you would need the formation of carbon polymers, and they think that this possibly occurred in the deep sea vents because it has a lot of heat and reducing chemicals like iron sulfide, and that this is how we got the first fatty acid chains to form, those first nucleic acid polymers and amino acid polypeptide chains, right? And that cells, the first thing on the outside of them is those membranes. So we had to have the formation of those phospholipids, which are, after they're created, are going to automatically form into those bilayers because energetically they want to do that. And then lastly, we had to have the development of some sort of mechanism of inheritance, and possibly that was RNA first. Okay, and again, evidence for that theory is that all organisms, we use the same 4 bases in DNA, all the A's, G's, T's, and C's. Our genetic code is universal. All organisms use the same 20 amino acids in their proteins, and we all use the same 64 codons to get there. So this suggests that there was a single ancestor for all of life on Earth. All right, so moving on to endosymbiotic theory. So the first organisms were clearly prokaryotes. And since you guys have been drawing these, you know that prokaryotes are structurally much simpler than other kinds of organisms, right? So how do we go from something very small and simple like a prokaryote to the craziness that we have in the plant and animal cells that have all of these compartments? And the idea is, sorry, let me just tell you what you're looking at here. So this is a macrophage. This is a eukaryotic, this is human cell, and it is chasing down a bacteria. So this is an E. coli that's just been ingested. And so this is like a white blood cell, and these are red blood cells. And looking at the red blood cells, what can you tell me about the hypertonic, isotonic, or hypotonic state of the solution? You should be able to tell me that. All right, so what we know is that eukaryotes, and you can see that in this picture, are much larger than prokaryotes, and that eukaryotes have compartmentalized organelles. It's the main difference between the two domains. And so then the question is, okay, how did eukaryotes get mitochondria and chloroplasts in the first place? And they think it happened kind of by like what you're watching right here. It was probably by somebody eating their neighbor, right? So this theory was first proposed by Lynn Margulis in 1981, and her idea is a pretty simple one, is that one prokaryote engulfed a second one, and that this increased its energy making capacity, which formed a beneficial relationship between the both of them that ended up becoming naturally selected for. And that this event occurred twice, once for mitochondria, which happened first, and then the second time for chloroplasts. So this was in a eukaryote that already had mitochondria that decided to engulf a cyanobacteria. And again, remember back in taxonomy, I told you that animals appeared before plants, and this would be why, okay? Because chloroplasts were not on the scene until after eukaryotes first came into existence. All right. So how did she get to this theory in the first place? So she studies amoeba. And this amoeba, she's watching them engulf other organisms, like this poor paramecia is about to right here. And she notices that the amoeba divides because it's a single cell and it's just going to divide and it makes two cells. But when she was watching the mitochondria inside this amoeba, she notices that these mitochondria divide independently and separately from the amoeba dividing. In fact, that suggests that they're able to function independently like a prokaryote. And in fact, they look quite a bit like them, right? So if we just line up some pictures here for you. One of these is a prokaryote, one of these is a chloroplast, and one of these is a mitochondria. So I'm going to just put this like 1, 2, 3, and you tell me who's who, right? Okay, so this can be kind of difficult, right? So this one is a mitochondria, and you can tell because it has the cristae going from left to right. And this one has the chloroplast, and you can tell that because the lines are kind of moving lengthwise. But this one is a prokaryote. And so first off, these things look very similar to one another. And then the other thing is, so when she gets in there, she starts looking at it, she's realizing that they both divide by binary fission, right? So mitochondria and chloroplasts, when they're dividing, they look a lot like just regular cell division. It has the same steps in binary fission as a prokaryote. They each have a double membrane, even though the prokaryote will have that extra cell wall, again, because it is free living and it needs to protect itself. But what really brought it home for her is when she was isolating mitochondria and chloroplasts, and she finds that they have their own DNA that is different from the eukaryote that was carrying it. Not only that, it's a single circular chromosome, just like the prokaryotes have. And then what brought it home was that the mitochondria and chloroplasts, just like prokaryotes, have their own separate protein-making machinery. They have 70S ribosomes. Remember, these are different than the 80S ribosomes that you find in a eukaryotic cell. So all of that suggests that these organisms were related to each other. They look alike. They have similar DNA chromosomes. They have their own protein-making machinery, and it's all the same kind. So if we go with the idea that one prokaryote ate another one and then for some reason it just didn't digest it, that would be like you eating a hamburger and you deciding to keep the hamburger alive in your stomach for like no reason. So why would you do that? The answer to that question is if dinner generates more energy alive than digesting it, you would too. So let's dial it back to the carbon cycle. And I know that feels like a lifetime ago. Remember that there were two kinds of respiration. So there's aerobic respiration and anaerobic respiration. And remember that aerobic respiration is with oxygen. So some bacteria use aerobic respiration, and all of our producers and consumers use aerobic respiration. And remember, there's anaerobic respiration, which was done by the methanogens. So again, remember, they love hostile conditions. So no oxygen, that soggy land that's very acidic, the methanogens really thrive in there. So no oxygen for our anaerobes. And so again, there's two types of respiration: anaerobic, which is no oxygen. And so what I didn't tell you before is that anaerobic respiration generates only 2 ATP molecules per glucose that it breaks down. And again, just remember that there was no oxygen on early Earth, so that was just fine for the bacteria that were there. But about 2.5 million years ago, cyanobacteria show up. So those are the first organisms that start doing photosynthesis, and they produced heaps of oxygen that actually ended up being toxic for the anaerobes, and it killed a lot of them. I think this was the largest extinction event on Earth. It killed 90 to 99% of living organisms at that time. And after there was so much oxygen in the atmosphere, a new organism comes around and starts doing aerobic respiration, which is using oxygen. But to add insult to injury, it was actually way more efficient at generating energy. So instead of having 2 ATP energy molecules per glucose, they were able to generate 36, which is like what, 18 times higher. So if you imagine that you're an anaerobic bacteria, and you see this jerk face aerobic bacteria nearby that's generating all this ATP, and you decide to eat said obnoxious aerobic bacteria, not only do you gain those 36 ATP if you keep it around, but now you have the ability to make ATP in both aerobic and anaerobic conditions. And so you better believe that your cells right now have this ability sitting there with you right now as you speak. So right now you're probably generating energy primarily through aerobic respiration. But you runners out there, you athletes, what happens when you run a lot? You know, you start to like run out of breath, and maybe you do it a lot, your muscles are going to get sore, right? So they get sore because your anaerobic respiration started to take over and produce lactate, which is getting in your muscles. So you use both of these quite often. So endosymbiosis in a nutshell, so you start with two independent bacteria, one of them engulfs the other, and when you have one bacteria now living inside the other, this is beneficial because you have just gained a huge amount of ATP that you did not have before. So that's known as mutualism. So the bacteria inside benefits because it's being protected and it's getting a lot of, I guess, other benefits from that organism. And so remember, this happened a second time. So cyanobacteria are just sitting around out there doing their photosynthesis thing. They're making glucose, and this whole process repeated again to make chloroplasts. So if you're a cell and you have your 36 ATP extra boost from eating that first one, why bother going out to get food if you can just go inhale your neighbor cyanobacteria who's going to make all of your sugar for you? So this is how plants evolved and came on the scene. And so again, just a reminder of evidence of that this actually occurred is that all three of these divide by binary fission. They each have a double membrane, and they each have their own single circular DNA chromosome, which is the big giveaway here that you're looking at a prokaryote. And again, they each have those 70S ribosomes. These, even when they're in a cell, this is independent protein synthesis of the rest of the cell. Okay, so that is all for today. Check the choice board for your extension activities. You do have to do that exit ticket and the Quizlet. So let me know if you guys have any questions. We is here for you. We're full service. All right, that's it for now. Take care.